In this last video for lecture 6, I just want to take a minute to talk about why it is some phenotypes in a population, even though they could arguably have a detrimental impact on individuals, don't end up knocking themselves out of the gene pool. Or put in other words, why it is that certain phenotypes get passed on from one generation to the next, even if they could have a negative impact on individuals. In the natural world, this doesn't really happen very often. It's kind of unique to humans. So let's think about something like this killer whale here. Killer whales are traditionally going to be black and white, but you can see this one has some sort of pigmentation abnormality going on. Killer whales should have what's called counter shading. This works for predators and for prey in the marine environment. They have a black top and a white bottom. And that means, if I am the prey of this killer whale and I'm swimming above it, looking down at the seafloor, the killer whale's black back is going to blend in with the seafloor and I'm not going to see it. Or if I'm swimming below the killer whale looking up at the sunny surface, I'm going to see the white belly of the killer whale and it's going to blend in with the sun and I'm not going to see it. And so the predator can sneak up on the prey. If this killer whale tried to do that, it's not going to blend in as well as its other members of its pod. So it's not going to be as successful at hunting and it might not live as long. It might not pass on its genes. Same thing goes for prey. Like these penguins here with this counter shading mutation are probably going to get eaten before they're able to reproduce, before they're able to pass on the mutations that gave them this abnormal coloration pattern, because they're not going to be able to blend in the marine environment. So their predators are going to pick them off easier than a penguin that has counter shading. In humans, like I said, oftentimes we see deleterious mutations or mutations that are harmful to individuals persist from one generation to the next, even though they are harmful. And I want to take a minute to talk about why that is. So one example would be something like Huntington's disease. I'm not sure if you are familiar, but Huntington's disease is a neurological condition that is fatal. And so a person who has a single allele for Huntington's disease, because it is dominant, so you only need one copy to get it, if they carry the allele for Huntington's, it will kill them at some point in their life. But Huntington's disease symptoms don't arise until mid to late in life, after people have already had children, after people have already passed on these genes. And so there are a lot of conditions like that where it might end up being fatal to a person, but they will have already passed on their genes to the next generation before they know they carry this condition. Another example for that would be early onset Alzheimer's. So if you've seen the film Still Alice, that covers this condition specifically, but same thing as Huntington's. This disease, if you carry the allele for it, will end up being fatal to you, but it often doesn't start until you're in your 40s, 50s, or 60s, after you've already passed on your genes to the next generation. And something that's kind of unique about this moment in time is in the past, even 20, 30 years ago, you wouldn't really have any way of knowing that you had this condition before it started to affect you. You wouldn't really seek out just a test to know whether or not you have this, and so you wouldn't know until you were already getting sick, until you'd already have kids. Nowadays, you can get genetic testing done through suppliers like 23andMe, and they can tell you when you're 20 whether or not you carry this gene, and so it can give you a lot more information to work with going forward. In this film, Alice has 3 children who all make different decisions about wanting to figure out whether or not they carry the gene that their mother had that killed their mother. So if this kind of topic interests you, I recommend that film for that reason. Another example that I think is perhaps more interesting, especially from an evolutionary perspective, is not a disease that doesn't start until later in life, but a disease that can hide in heterozygotes. You might recognize this individual here is Prodigy from Mobb Deep, and Prodigy died a few years ago from complications due to sickle cell anemia. Sickle cell anemia is a recessively inherited condition, and so an individual like Prodigy had 2 copies of the recessive allele. He had sickle cell anemia. He made hemoglobin molecules that were an abnormal shape, which meant his red blood cells were sickle-shaped instead of platelet-shaped. People like Prodigy would have been, okay, they used an R here, I used an A, but again, lowercase means recessive. So people like Prodigy who have sickle cell anemia would have had 2 copies of the recessive allele. They would be homozygous recessive. People who do not have sickle cell anemia and do not carry a allele for the condition would be homozygous dominant. Anybody who is a heterozygote for sickle cell anemia would have 1 copy of the unaffected allele, big R, and 1 copy of the affected allele, little r. And carriers can literally carry this trait from one generation to the next. And so we can say a heterozygote carrier is an individual who does not have a condition, So big R, little r. They don't have sickle cell anemia, but they carry the allele for sickle cell anemia. And so you can imagine, like these two parents here, these two parents are both carriers, big R, little r, big R, little r. And then when they have children, they could have children who are completely unaffected and not carriers. They could have children who are affected with both copies of the affected allele, or they can have children just like themselves who are also carriers. And so there are conditions like sickle cell that can persist in populations over generations just because individuals who are carriers can take it from one generation to the next without necessarily being negatively affected. Okay, and I want to take a little time to talk about sickle cell specifically because it is a really sort of unique example. If we think about the incident rate of sickle cell anemia in the U.S. specifically, the most recent data we have is from about 2010. But if we look at the number of cases of sickle cell per 1,000 babies of various races, then we're going to see a larger proportion of individuals that have sickle cell in the Black community, and specifically people of African descent, than we see in any of these other scenarios. And I want to talk about specifically why that is, why we would see a phenotype that is more prevalent in one group than another. And so we've talked so far about alleles that are deleterious, alleles that are having a negative impact on individuals. But alleles can also have hidden benefits, and that's exactly what is happening here. Alleles can give individuals a leg up in the race to survive, in the race to pass on genes. And so if we look at, I said specifically of African descent here, that's important. If we look at the continent of Africa in terms of the breakdown of people that carry the sickle cell allele versus the continent of Africa in terms of the distribution of malaria, we can see there is a large percentage of the population that carries the sickle cell allele all across the equatorial African continent, in line with this part of the continent that is high prevalence for malaria. And just like we talked about earlier in ways that phenotypes can differ on a spectrum due to influences from the environment, that's exactly what is happening here. Ecological problems such as malarial infection have selected for sickle cell anemia in this population. And this is true for anywhere in the world that has high malarial prevalence. It's especially true for equatorial Africa. And this is because heterozygotes, individuals who are carriers for sickle cell but don't have the condition, are at an advantage against malaria. They aren't able to get a malarial infection like somebody who is homozygous dominant would. And what I mean by that is, if we look at the blood cells themselves, somebody who is homozygous dominant has platelet-shaped red blood cells, like you see here. So if you do not have sickle cell and you do not carry sickle cell, all of your blood cells look like this. Somebody who has sickle cell anemia, like Prodigy did, all of his blood cells look like this. They are sickle-shaped. And if you are a carrier, remember carriers express both types of alleles, so they express both of the proteins, the hemoglobin that is clover-shaped and the hemoglobin that is oblong. They're going to create mostly red blood cells that are platelet-shaped, but also some red blood cells that are sickle-shaped. Not that many, but a couple. And what happens if you get bitten by a mosquito that has malaria is if you are this genotype, if you have platelet-shaped red blood cells, the malaria is going to go inside your red blood cells, reproduce in there, and you will have a full-blown malarial infection. If you have sickle cell anemia, malaria can't fit inside those red blood cells, so you will not be infected by malaria. People who have sickle cell anemia are resistant to malaria, and so are people who are heterozygotes, people who are carriers. Because somebody who's a heterozygote, if a mosquito with malaria bites them, the malaria is going to start to try to reproduce inside of these red blood cells, but as soon as a hypoxic condition is reached, as soon as oxygen levels drop, this person's blood cells are going to switch from platelet-shaped to sickle-shaped. And so it's basically going to induce the sickle-shaped phenotype, which makes them completely resistant to malaria, and the infection will clear. And so what this meant through time is that in modern medicine, we have ways to treat sickle cell so people can live long and full lives. We have ways to treat malaria so people can live long and full lives. But evolutionary speaking, that's only a very recent advancement in medicine, certainly within the last sort of 50 to 100 years. So before that, if you were living in a malaria region and in a region with sickle cell, individuals with sickle cell, 2 copies of the recessive allele, would die in childhood due to sickle cell attacks. So they would die before they could grow up and pass on those genes. And individuals without sickle cell and who were not carriers would die from malaria, usually before reproductive age. And so we were knocking people out of the gene pool if they were homozygous. If they were homozygous dominant because they were dying from malaria, or if they were homozygous recessive because they were dying from sickle cell anemia complications. And only heterozygotes were most likely to survive into adulthood and pass on their genes. And so they would pass on one copy of the dominant allele and one copy of the recessive allele. And it would mean they would not suffer from sickle cell attacks, and it would also mean they would not die from malaria. And if we want to go ahead and look at a blood sample here, this is a blood sample from a person right here. This is a blood sample from a person who is homozygous dominant for the sickle cell gene. So they have platelet-shaped red blood cells. This is a red blood cell. And all these little like weird squiggly things, these are malarial parasites. And they literally, if you get infected with malaria, it's a blood infection. And so these parasites burrow into your red blood cells, they lay eggs in there, and then the red blood cells burst and the infection spreads to other red blood cells in the bloodstream. So it's really bad news bears if you are a person that does not have sickle cell anemia or does not have at least carrying the gene for sickle cell anemia if you were to get infected with malaria. All right, and then lastly, to kind of wrap up this lecture, I want to talk about one more example. In everything we talked about so far, we've sort of said, okay, if you have this allele, you have the condition. If you don't have this allele, you don't have the condition. But there are other scenarios in which you can carry certain alleles, and it doesn't necessarily give you something or not give you something, but it does put you at a higher risk or put you in a risk category for certain phenotypes. And an example of that would be breast cancer and the BRCA genes. You might be aware of this if somebody in your family has ever had breast cancer, or perhaps you yourself have been tested for these genes. BRCA genes, BRCA1 and BRCA2, these genes are tumor suppressor genes. So these genes code for information that makes tumors less likely to develop and less likely to grow. And you have 2 alleles for every single one of these genes. And so the majority of the population has 2 alleles for BRCA1 and 2 alleles for BRCA2 that work just fine, that make tumor suppressor proteins that help tumors not to grow or not to proliferate. But if you have a mutation in the BRCA1 gene for one allele, or in the BRCA2 gene for one allele, or in the BRCA1 gene for both alleles, or the BRCA2 gene for both alleles, then that puts you at a much higher risk for developing cancer because now you have an error in the machinery that should check for and prevent tumors. And so a lot of people who have a history of breast cancer in the family will now get tested for BRCA mutations as well. And somebody that did that is pictured here, Angelina Jolie. Angelina Jolie, when she got her BRCA test, she found out that I think she had 3 of the 4 possible mutations within the BRCA genes, between BRCA1 and BRCA2. And having those 3 mutations put her at a much, much higher risk of developing breast cancer and ovarian cancer in her lifetime. So she chose to have an elective and preemptive double mastectomy and oophorectomy. So she had her ovaries removed and her breasts removed. In the general population, about 12% of women will develop breast cancer over the course of their lives, but women with inherited mutations in the BRCA1 and BRCA2 gene put that risk up to 60%. And if you have additional mutations beyond just one in those gene regions, it puts you even higher than that, like Angelina was in. So again, in the kind of unique time we find ourselves in, people can find out a whole lot more information about the information that they carry in their genetic in their genomes, and what that means for them as individuals as they make choices going forward about having children, or what that means for them as individuals as they make choices about their potential futures, or what that means for them as individuals if they have mutations in these pathways and they want to preemptively prevent any sort of cancer development. And if this is something that really interests you, then you can think about or look into a career in genetic counseling. A lot of couples nowadays will engage in genetic counseling prior to trying to conceive, just to kind of figure out what are the options going forward, what are the probabilities going forward. If I were to have kids with this person, what combinations of alleles are possible, which of those are potentially detrimental, and what can we do to prevent any sort of future problems that might be associated with BRCA1 or BRCA2 for myself as an individual. So if this is something that you want to look into, I definitely encourage it. Or if you want to talk more about this, we can definitely do that over email.